Hello, Mr. Allman here, and this is your Almondcast on enzymes. Enzymes are proteins that have the function of being able to catalyze chemical reactions within the body. Chemical reactions are happening all the time at an astounding rate, and it's because of the work of the thousands of different enzymes within each of your cells that we are able to process energy and stay alive. Let's get into it. First, a little bit of theory in terms of understanding the connection between chemistry and physics to what's happening in our cells. First, remember that matter and energy cannot be created or destroyed. Matter and energy can be transformed, and that's ultimately what we see happening here. As organisms, we are always processing energy. We know that producers are taking energy from the sun. We know that as heterotrophs, we as animals are always eating, we're consuming food and being able to extract energy from that. We're using that energy to be able to do work, whether it's to produce heat, to move, or to maintain homeostasis. Now, there are two types of chemical reactions. There are those that are exergonic. Exergonic reactions are going to release energy. So you can see that the energy present in the reactants is higher than the energy present in the products. So the difference there is the energy that's going to be released. Now, in chemistry, sometimes we'll interchangeably use the word exothermic, but heat is not necessarily the type of energy that's going to be released from these reactions. Quite on the contrary, in biological systems, cells want to actually harness that as chemical energy as opposed to harnessing it as heat. Catabolic reactions, those that divide molecules, are going to be exergonic. So think of what's happening during the process of digestion or during the process of cell respiration that is going to release energy. On the contrary, endergonic reactions are ones in which the reactants have a lower amount of energy than the products. Often that's because the products are going to be larger molecules that have more chemical bonds in them, and so additional energy is required to be able to build those. So energy storage is going to be a great example of an endergonic reaction. Being able to take, for example, excess sugar that's present in the blood and combine those sugar molecules to make a polysaccharide called glycogen. Other important endergonic reactions are like photosynthesis because of the complex sugar molecules that are made from carbon dioxide and water, and the process of building big molecules like proteins or DNA. A second thing that's important is that any closed system, the universe, an ecosystem, a community, is going to move towards entropy. So maximum entropy is going to be evening everything out. It's going to be going against what we see in life. Life is highly ordered, and so energy is required to be able to maintain that order. An important molecule that's used as a way to be able to transmit and convert that energy is ATP. ATP is adenosine triphosphate. We would classify this as a nucleic acid in terms of what type of organic molecule it is. This is being used in huge amounts in all of our cells, and it's being used continuously. It's made up of a nitrogenous base, a ribose sugar, and 3 phosphate groups. So you can see the P stands for phosphorus, and so this is, again, in the nucleic acid category. That third phosphate on the end is relatively unstable. The bond that holds that final cluster of phosphorus and oxygen atoms there, it is relatively easy to be able to break. And by easy, I mean it doesn't take a lot of energy. The input of energy is fairly small to sever that covalent bond, that phosphate bond there, and meaning that as it's released, there's going to be more energy available to be able to do some other work within the cell. A couple examples of that is ATP being used with transport proteins during active transport. So in active transport, we're trying to move a molecule against its concentration gradient. So if I'm trying to transport this particular solute, so it's called a sugar molecule, into the cell, ATP is required to be able to cause the change in shape of the protein. So you can see that as ATP binds, the phosphate connects to this protein, and the energy from that phosphate, from the breaking of the ATP, is going to change the shape, release that particular molecule out, change the shape of the protein to be able to then release the solute into the cell. So this is a way to be able to create a concentration gradient, and ATP is the energy necessary in active transport. Another example in the idea of movement, a motor protein is the type of protein that moves along the cytoskeleton and drags a vesicle. You should remember these from our cells unit because vesicles, remember, are the ways in which molecules can be transported between the Golgi and another place like the lysosome or the plasma membrane. So as ATP binds to the motor protein, the phosphate, that third phosphate, is cleaved off, and the energy from that exergonic process is going to cause a change in the shape of the motor protein and drag that vesicle in the direction that it needs to go. All of this is helpful for the process of metabolism. So in metabolism we have the connection between exergonic energy releasing processes, particularly cell respiration or fermentation, and the work that's done to be able to build those things up like photosynthesis. Now, all of this leads us into enzymes, and specifically how it is that enzymes impact all of these reactions happening within the body. So enzymes, as I said, are catalysts, and this type of chart is something you should be familiar with being able to interpret. You can see the energy present in the reactants, so higher up on our vertical axes means more energy. So in this case, the products are at a lower energy, probably because they are smaller and contain less bonds than the reactants were. So the process of going from reactant to product is going to release energy. However, that process isn't going to happen without what's called the activation energy, because before new bonds can be formed and that energy can be released, some energy input is required to get that reaction started and to move them towards what's called a transition state. Now, if there was no enzyme present, the amount of activation energy is much, much higher, represented by the black line. And what enzymes do is that they affect the shape of the substrate and lower the amount of energy that's needed to break those initial bonds. So that lower activation energy is going to allow the reaction to progress, and often to progress more quickly than it would otherwise. Usually what would happen in reality is that if there is an enzyme, the reaction would occur because enough activation energy is present in the cell based on the amount of heat that our cells are warmed to. If there was no enzyme, there would be no reaction at all. So if the enzyme is lost, if the enzyme was never there to begin with, or if the enzyme is destroyed for some reason, the reaction is going to cease completely. You should remember enzymes based on the ASE ending, which is the common naming system that's used for enzymes. And the prefix is going to be the enzyme substrate. So lactase is going to, the ACE at the end means it's an enzyme, and lactose is the substrate. So it's named after what the enzyme is actually working upon. So enzymes interact with the substrate. The reactants, in this case if we have two reactants that are changing form, they are going to join with the enzyme at a place called the active site. Once they join at the active site, there could be some small shape change, or something is happening to the substrate by the enzyme when it's attached there that's going to lower that amount of activation energy. When those things are combined, we call that the enzyme substrate complex. Then once the reaction is complete, the original substrates, now that they have been changed, are released as products, whether they have been joined together, whether they've been broken apart, or simply changed in some way. Enzyme substrate specificity describes the relationship between the active site and the substrate to which it will bind. Shape is important in that, as well as size. Those are the physical properties that determine whether or not the substrate will physically fit into the active site, as you see here. Those things need to physically join together to form that enzyme substrate complex, which will then lower the activation energy to cause the reaction to occur. Additionally, chemical properties need to match. So the substrate needs to have some type of chemical attraction towards the active site. If the chemical properties don't match, the substrate could be repelled. For example, if the substrate is positively charged, the active site of the enzyme should have a negative charge to it to attract that substrate towards it. The active site is localized to just a small area on the enzyme, so the whole enzyme's shape isn't necessarily important, only the shape of the active site. A change in the shape of the protein away from the active site may or may not actually impact the ability of the enzyme to complete its job. On the topic of organization, first remember that enzymes are proteins, and proteins can be transported to a particular spot. As they're made on the ER, they can then be packaged in vesicles and sent to a place like the mitochondria, to the nucleus, to the plasma membrane. And so specific enzymes, because they are responsible for specific steps in a chemical reaction, they can be made and sent to that one particular place, and that makes the process more efficient. If you're going to have 1,000 different enzymes within a cell, it's not going to be efficient if those 1,000 different types of enzymes are scattered everywhere. Rather, if we have enzymes responsible for the process of respiration, because respiration happens in the mitochondria, you will find enzymes being sent only to and being found only in the mitochondria. The rate of enzymes is going to be impacted by environmental conditions, and the enzymes of digestion are a great example of this. You can see how pH is one of the factors that impacts the rate of reactions. In the stomach, you have certain enzymes like pepsin that are responsible for breaking down protein. Whereas in the intestine, you have other enzymes such as trypsin, which are also responsible for breaking down protein. Now, each of these two enzymes has a similar function, but they're located in a different part of the body. One in the stomach, one in the intestine. The intestine has a fairly basic pH with a pH around 8, whereas the stomach has a pH near 2 because of the presence of stomach acid. So each of those enzymes is going to fold up into its three-dimensional shape based upon the environmental pH that is necessary. If you take that enzyme out of that optimal pH, it's probably going to denature, and when it denatures, it's going to lose its function. So if I take stomach enzymes and put them into a neutral pH, they're going to unfold, and as a result, the protein is going to lose its active site and no longer function. Temperature impacts it because temperature is what allows for that activation energy to be able to get the reaction going. So you can see that there are heat-tolerant bacteria that will still be able to have enzymes that perform at extremely high temperatures, whereas the temperatures that we see in our body, like human enzymes, are going to be optimally work around 37 degrees. You'll notice that if you increase the temperature past about 42, 44, 45 degrees, the activity of those enzymes drops off dramatically. And so you can imagine what would happen with a severely high fever when you get above 42, 43 degrees Celsius, why it is that that's a pretty serious situation that is definitely capable of causing a person to die. If you stop all the biochemical reactions within cells, then their ability to process energy and their ability thus to stay alive is going to end. Concentration is also important. If you increase the amount of substrate, as that substrate concentration increases, you will increase the rate of the reaction up to a certain point because there's a limited number of enzymes. So as you have more substrate, you make it more likely that enzyme and substrate are going to find each other. If you flood the reaction or flood the cell with more and more and more substrates, you are limited by the number of enzymes. So having more substrate past that saturation point isn't necessarily going to speed up the reaction anymore because you're waiting for the enzymes to actually be able to catalyze the reaction. For all enzymatically controlled reactions, there are ideal conditions. There's an ideal temperature, an optimal pH, and an optimal substrate concentration, which will allow a particular reaction to happen more quickly than it would otherwise. Finally, this is an HL topic, and that is how enzymes are going to be regulated. There are competitive and non-competitive inhibitors, and each of those are working to be able to slow or stop the rate of a reaction within a cell. In competitive inhibition, a molecule other than the substrate, so a competitor, is going to bind to the active site. Because the competitor is bound to the active site, the substrate, represented by the green blob here, is no longer able to bind, and so the reaction does not occur. If you have a non-competitive inhibitor, it's going to bind to an allosteric site. So the allosteric site is here on the bottom of the enzyme. This is not the active site. However, the presence of that non-competitive inhibitor is going to prevent, again, the substrate from binding, and so the reaction will not occur. The way that is going to occur, that inhibition is going to occur because it's going to change the shape of the enzyme, particularly the shape of the active site. As you can see on the bottom here, when the inhibitor is bound to the allosteric site, the active site shape is changed, and so the substrate can no longer bind. Whereas you can actually have the opposite case. We can have activators that can also bind to an allosteric site and cause the enzyme's active site to be opened. So the presence of these additional other molecules, these other molecules will be able to regulate whether or not the enzyme is on or off, so thus whether a reaction can happen or whether a reaction is stopped. This is important because of the topic of feedback. So feedback is the way that regulation can stop unnecessary reactions from occurring. So for example, we have the substrate threonine. So threonine is one of our amino acids. Threonine, there is a series of reactions that can turn threonine into a molecule, another amino acid called isoleucine. So isoleucine is the product. Threonine is the original substrate. There's a series of steps each with their own enzyme necessary to get that final product. So it's a multi-step metabolic pathway. Now, if isoleucine is needed, then this reaction is going to occur. But what if a lot of isoleucine has already been produced and it's not needed anymore? If you have a lot of it floating around within the cell, it's going to waste a lot of energy, and it's going to waste more substrate for the cell to make more that it doesn't need. So, isoleucine is actually an allosteric inhibitor. It's a non-competitive inhibitor that binds to the original enzyme that started the whole chain of events. So if isoleucine is present, if there's extra of it present in the cell, it's going to bind to that original threonine deaminase enzyme. It's going to change the active site and make it so that the active site is no longer available, and it's going to stop the reaction. This idea of regulation is necessary because of the number of enzymes and the number of reactions happening. We need some way to be able to control when it is that a certain reaction is going to happen, so that it will happen when it's to produce a product that's needed, but it will be turned off when a product is no longer needed. That brings us to the end of our Almondcast on enzymes. Please let me know if you have any questions. Thank you.